Doc.
Good
So
morning,
let's
Doc.
start. It's already 10:30. Can you see the slides now?
Yes, Doc.
Okay, so this morning we'll talk about muscle biochemistry and glycogen metabolism. But let's start first with muscle biochemistry. So I know you are familiar with this figure. Basically, this is the schematic representation of the thin filament showing the spatial configuration of the three major protein components, which is your actin filaments. You also have your G actin and the F actin, your troponin, as well as your tropomyosin. So basically, your monomeric G actins makes up about 25% of the muscle protein by weight. So at physiologic ionic strength and in the presence of your magnesium, so with magnesium, the G-actin polymerizes non-covalently to form this insoluble double helical filament, which is your F-actin. So basically, um, in the muscle, you have your tropomyosin and the troponin complex, which is present in the thin filaments, and this basically performs the key functions in striated muscle. So in striated muscle, there are two other proteins that are minor in terms of their mass, but also important in terms of their function. So you have your tropomyosin, which is a fibrous molecule that consists of your two chains, which is your alpha and the beta, which eventually attach to the F actin in the groove between filaments. If you try to look back in the previous figure. So you also have the troponin complex, and this is unique to striated muscle, and this consists three polypeptides. Now you have your troponin P, your troponin I, as well as your troponin C. So your troponin T binds to the tropomyosin as well as the two other troponin components. So basically, this is your troponin complex, and what binds the three troponin complex is your troponin T. That also binds your tropomyosin. On the other hand, your troponin I inhibits your F-actin myosin interaction, which will be discussed later, and this also binds to the other components of the troponin. And lastly, you have your troponin C, which is a calcium binding polypeptide that is structurally and functionally analogous to your calmodulin, which can bind up to 4 calcium ions per molecule of your troponin C. So if you recall, your calmodulin is an important calcium binding protein that is widely distributed, no, in the body. So this one is your major proteins of the muscle. This is a diagram of a myosin molecule showing the two intertwined alpha helices. So you have your alpha helices that is your fibrous portion, and you also have this globular region or the head, the light chains and the effects of the proteolytic cleavage by the trypsin and your papain. No? So your myosin contributes about 55% of the muscle protein by weight and forms the thick filament. So when your myosin is digested with trypsin, so basically your trypsin acts on um, the fibrous component, the two myosin fragments, which are your meromyosins, are being generated. So you have your light meromyosin and your heavy meromyosin. So this is basically digested with the use of your or the help of your trypsin. So your light meromyosin consists of the aggregated insoluble alpha helical fibers that is from the tail of your myosin. So this exhibits no ATPase activity and does not bind to your F-actin. On the other hand, you have your heavy meromycin, which is a soluble protein that has both a fibrous portion here and the globular portion. So this basically exhibits ATP activity and eventually binds to your F-actin. So when this is digested, your heavy meromycin is digested by your papain, this eventually generates two subfragments, which is your S1 and your S2. So the S2 fragment is fibrous in character, and this has no ATPase activity, just like your light meromycin. While your S1 does exhibit your ATPase activity. This eventually binds to your L chains, and in the absence of ATP, will bind to and decorate the actin with your arrowhead. So basically, both S1 and your heavy meromycin exhibit ATPase activity. So if you try to deduce this one, basically the ATPase activity resides in the S1 or the globular head or the globular portion of your heavy meromycin. Now let's proceed with the hydrolysis of ATP. Basically, the hydrolysis of your ATP will be responsible or will drive the association as well as dissociation of both your actin and your myosin filament. No? And this involves five reactions. So I have to refer you back to the figure that was present in the previous edition of the book, because this basically describes a major biochemical event occurring during one cycle of your muscle contraction and relaxation. So what happens is that, no, in the relaxation phase of your muscle contraction, the S1 head of the myosin um, hydrolyzes your ATP to ADP. So when this happens, diba, you have your ATP myosin complex, and when this ATP is being hydrolyzed to ADP and your PI, there will be a resultant complex, which is your ADP-PI myosin complex. So this um, product remain bound. No? However, when you have your ADP myosin, PI myosin complex, this has been already energized, and this is the so-called high energy configuration or conformation. Now, when contraction of the muscle is being stimulated, and this is brought about via the events that involves your calcium, your troponin, your tropomyosin, and your actin here becomes accessible. And the S1 head of the myosin finds it, binds it, and eventually forms your actin myosin ADPPI complex. So this is the product of the second biochemical event that occurs during one cycle of muscle contraction and relaxation. So the formation of this complex promotes the release of your PI first. Once this PI is released, that initiates the power stroke. So this is followed as well by the release of your ADP. And this is accompanied by a large conformational change in the head of the myosin in relation to its tail. So basically, it pulls the actin about 10 nanometers towards the center of the sarcomere. So this is now your power stroke happens. So the myosin here is now in a so-called low energy state. And this is indicated by the actin myosin complex. So basically, you have to remember that what initiates the power stroke is the release of your PI. Now, when This is followed by the release of your ADP. Now there will be a conformational change in the head of the myosin. This is basically where your low energy state becomes the product of this biochemical reaction. Now, another molecule of ATP binds to the S1 head that eventually forms your actin-myosin ATP complex. So once you have this as the product of your biochemical reaction number four, no, the incorporation of your ATP or the binding of your ATP to the low energy state, which is your actin myosin complex, this forms the actin myosin ATP complex. And when you have this, your actin myosin ATP has low affinity for your actin. So what happens is that your actin is being released, and this is your last step, which is the key component in the relaxation of your muscle. So basically, this is dependent upon the binding of ATP to the actin myosin complex. So what drives the relaxation is the binding of your ATP to your actin myosin complex. And because this actin myosin ATP complex has uh, low affinity for your actin, the actin is being released and your muscle relaxes. So this is basically the five biochemical reactions that happens in the um, muscle contraction and relaxation. So basically, this is what is the important part of your um, reaction. So the hydrolysis of ATP is used to drive the cycle, with the actual power stroke being the conformational change in the S1 head that occurs upon the release of your ADP. So now when your intracellular levels of ATP drops, like when death happens, your ATP will now be unavailable to bind to the S1 head. No, in the step four of the reaction, actin does not dissociate, so it remains bound, and relaxation does not occur. So this is what happens during death. No, there is stiffening of the body that occurs after death. So this is basically your rigor mortis. So when we talk about regulation of your muscle contraction, there are two general mechanisms. An actin-based, which is unique to your skeletal and cardiac muscle, and myosin-based for your smooth muscle. So this figure is a diagram showing to us the relationships among the sarcolemma, no? The plasma membrane, your T-tubule, and the two cisternae in your sarcoplasmic reticulum of the skeletal muscle. So you know that the T-tubule extends inward from the sarcolemma. So when there is a wave of depolarization that is initiated by a nerve impulse, it is then transmitted from the sarcolemma down to the T-tubule. So this is conveyed to the calcium release channel or your RYR channel, perhaps by the interaction between it and the dihydropyridine receptor, which is your slow calcium voltage gated channel. So the release of calcium from your calcium release channel into the cytosol will be the one responsible for initiation of contraction. Subsequently, your calcium is pumped back into the cisternae of the sarcoplasmic reticulum by your calcium ATPase or your calcium pump, and stored there in part bound to your calsequestrine. So there are events that leads to the opening of your RYR receptor, and this is the possible chain of events that leads to the opening of your calcium release channel. So the calcium, your After the depolarization of the nerve, there will be depolarization of the skeletal muscle, and then depolarization of your transverse tubular membrane or your T-tubule. So after that, there will be a charge movement of the slow calcium voltage channel of the transverse tubular membrane. And this is what is thought to be responsible for the opening of your calcium release channel or your RYR1 receptor. So going back in the sarcoplasm of a resting muscle, the concentration of your calcium is about 10 to the negative 8 to 10 to the negative 7 moles per liter. And the resting state is achieved because your calcium is pumped back into the sarcoplasmic reticulum through the action of an active transport system, which is your calcium ATPase um, calcium ATPase that is responsible for the initiation of the relaxation. So inside the sarcoplasmic reticulum, your calcium is bound to a specific calcium binding protein that is designated as your calsequestrine. And the sarcomer is surrounded by an excitable membrane, which is your T-tubule, composed of your transverse channels closely associated with your sarcoplasmic reticulum. So now let's move on to malignant hyperthermia. So basically, your malignant hyperthermia um, results from the mutation in the gene that encodes the calcium release channel. So this severe reaction happens on exposure to certain anesthetics like your halothane, as well as your depolarizing skeletal muscle relaxants, which is your succinylcholine that serves as the example. So this reaction consists primarily of rigidity of the skeletal muscles. There will be hypermetabolism and high fever. So the high cytosolic concentration of your calcium in the skeletal muscle is thought to be a major factor in the causation of malignant hyperthermia. So appropriate treatment when you talk about malignant hyperthermia that is brought about by your anesthetics or your depolarizing skeletal muscle relaxant is to stop the anesthetic and administer your drug, which is your dantrolene, that will be given intravenously. So this dantrolene is a skeletal muscle relaxant that acts to inhibit the release of your calcium from the sarcoplasmic reticulum into the cytosol, thus preventing the increase of cytosolic calcium that is found in your malignant hyperthermia. So this figure is a simplified scheme of the causation of your malignant hyperthermia. So as what has been said, there are many different point mutations that have been detected in the RYR1 gene, some of which are associated with central core disease. So it is estimated that at least 50% of the families with members who have malignant hyperthermia are linked to the RYR gene. There are some individuals with mutation in the gene encoding your DHPR have also been detected, and it is also possible that mutation in other genes for proteins involved in certain aspects of muscle metabolism will also be found. So what happens when there is mutation in the RYR genes is that there will be alteration of the calcium release channel protein, no? Basically, there will be substitution of your cysteine for arginine. And the mutated channels open more easily and stays open longer, thus flooding the cytosol with your calcium. So when you have high intracellular levels of calcium, they stimulate the sustained muscle contraction that causes rigidity. So your high calcium also stimulates the breakdown of your glycogen, glycolysis, as well as aerobic metabolism that results in excessive production of heat. That is what is basically the pathophysiologic cause of malignant hyperthermia. Now let's proceed with the cardiac muscle. So the general picture of muscle contraction in the heart basically resembles that of skeletal muscle. And the cardiac muscle, just like your skeletal muscle, is created and uses the actin tropomyosin-troponin system that has been described earlier. So unlike the skeletal muscle, no, cardiac muscle exhibits intrinsic rhythmicity, and individual myocytes communicate with each other because of its syncytial nature. So here, the T-tubular system is more developed, the sarcoplasmic reticulum is less extensive, and consequently, the intracellular supply of calcium for contraction is also less. So basically, the cardiac muscle relies on the extracellular levels of your calcium for contraction. Your cyclic AMP plays a more prominent role in cardiac muscle as it modulates the intracellular levels of calcium through the activation of your protein kinases that phosphorylate the various transport proteins in the sarcolemma and your sarcoplasmic reticulum. So, as stated above, the extracellular calcium plays an important role in the contraction of your cardiac muscle, but not in the skeletal muscle. So this means that your calcium both enters and leaves the myocyte in a regulated manners. And this is being regulated by the three channels. You have your calcium channels, your calcium sodium exchangers, and your calcium ATPs that has been mentioned early on. So your calcium channels, you have your L type and your fast or T type. No, the L type has long duration current and it has large conductance. This is also being referred to as your slow calcium channels. This is a voltage gated and is equivalent to the dihydropyridine receptors of the skeletal muscle. And this is basically regulated by your CAMP-dependent protein kinases, which is stimulatory to it, and your CGMP-dependent protein kinases that is inhibitory to your slow calcium channel. On the other hand, your fast or your transient calcium channels contribute to the early phase of the increase of your myoplasmic um, calcium. So basically, your calcium enters the myocyte via highly selective channels. And the major portal of entry is the L-type channels that stimulates your cardiac conduction, or rather cardiac contraction. So you also have your calcium sodium exchanger. So the energy for the uphill movement of your calcium out of the cell comes from the downhill movement of the sodium into the cell from the plasma. So this exchange contributes to the relaxation but may run in the reverse direction during excitation. So because of the calcium sodium exchanger, anything that causes your intracellular sodium to rise will secondarily cause your calcium to rise as well, causing a more forceful contraction. This is referred to then as the positive inotropic effect, no? The forceful contraction. One example is that of the drug digitalis. Your digitalis or your digoxin is used to treat heart failure. So basically, the role of your digitalis inhibits the sarcolemmal sodium potassium ATPase, diminishing your the exit of sodium by this route, thus increasing sodium ion. So this promotes the inflow of your calcium via the calcium sodium exchanger, and the increase in the calcium results in increased force of cardiac contraction that is beneficial in patients with heart failure. Now, we also have your calcium ATPase, which is situated in the sarcolemma, and this is responsible or contributes to the exit of your calcium from the sarcolemma. So this play a negative minor role, a relatively minor role rather, as compared with the calcium sodium exchanger. Now let's proceed with the smooth muscle. So your smooth muscles have molecular structures that is similar to those in striated muscle, but here the sarcomeres are not aligned so as to generate the striated appearance of your smooth muscles. So this contains your alpha-actinin and tropomyosin molecules, like just like your skeletal muscles do, and they do not have the troponin system. To take note, this troponin system is absent in your smooth muscle, and the light chains of smooth muscle myosin molecules differ from that of your striated muscle myosin. So the regulation of your um, excuse me, the relaxation of your the regulation of the smooth muscle contraction is myosin based, and this is also regulated by your calcium. So when there is a regulation, contraction rather, of your smooth muscle, there are no detectable ATPase activity when your smooth muscle myosin is bound to the F-actin in the absence of your other muscle proteins. So this absence, no, of the activity is quite unlike the situation described for striated muscle myosin and F-actin, which has an abundant ATPase activity. So your smooth muscle myosin contains the light chains that prevent the binding of the myosin head to the F-actin. So what happens is that they must be phosphorylated before they allow F-actin to activate your myosin ATPase. So the ATPase activity then attained by the hydrolysis of your ATP that is about tenfold more slowly than the corresponding activity in skeletal muscle. So the activity of your ATP in smooth muscle is slow. The phosphate on the myosin light chain may form a chelate with a calcium bound to the tropomyosin troponin actin complex, leading to an increased rate of formation of the cross bridges between your myosin heads and actin. So the phosphorylation of your light chains initiates the attachment detachment contraction cycle of your smooth muscle. So here, this figure shows us the regulation of your smooth muscle contraction by your calcium. So take note that your PL myosin is the phosphorylated light chain of the myosin, and your L-myosin is the dephosphorylated light chain. So in smooth muscle sarcoplasm, it contains the myosin light chain kinase that is calcium dependent. So the calcium activation of the myosin light chain kinase requires the binding of your calmomodulin and for calcium activated light chain kinase that phosphorylate the light chains, no? Which then cease to inhibit the myosin F-actin interaction. So when this L-myosin is being activated, it starts or the contraction. So the contraction cycle in the smooth muscle begins. Okay, so this table shows to us the actin interaction in striated and smooth muscles. So here, take note that the proteins of the muscle filament in striated muscle includes your actin, myosin, and tropomyosin complex, which is also found in your smooth muscle. However, your troponin is not part of the muscle filaments in your smooth muscle. When we talk about spontaneous interaction of the F-actin and the myosin alone, no, this is seen in striated muscle but not in your smooth muscle. So the interaction or the inhibitor of your F-actin myosin interaction is through your troponin system in striated muscle, and particularly your troponin I. While in the smooth muscle, you have your unphosphorylated myosin light chain. Both striated and smooth muscle contraction is activated by your calcium, and your effect of your calcium in the striated muscle is through binding of the calcium for calcium ions to the troponin C, no, that is responsible um, in the um, interaction of your calcium to the um, light chain. However, in smooth muscle, you have your calcium ion that binds to calmodulin, as what was shown earlier. So the effect of your protein-bound calcium, no, in striated muscle, your troponin C antagonizes your troponin I inhibition of the F-actin myosin interaction. So this allows your F-actin activation of your ATPase, while in smooth muscle, your calmodulin calcium activates the myosin light chain kinase that phosphorylates your myosin P light chain, and when you have your phosphorylated form of your P light chain kinase, this no longer exhibit or inhibits the F-actin myosin interaction, allowing your F-actin activation of your ATPase. So this is a diagram that shows to us the sources of your ATP in muscle. And we all know that ATP is required as the constant energy source for the contraction relaxation of your muscle. And this can be generated by glycolysis using your blood glucose or your muscle glycogen. It could also be through your oxidative phosphorylation or from your creatine phosphate, and lastly, from two molecules of your ADP in a reaction that is catalyzed by your adenyl kinase. So the amount of ATP in skeletal muscle is only sufficient to provide energy for the contraction for a few seconds only, so that your ATP must be constantly renewed from one or more of the above sources, depending upon the metabolic conditions. So there are at least two distinct types of fibers in skeletal muscle, one predominantly active in aerobic condition and the other in anaerobic condition. But not expectedly, they use each of the above sources of energy to a different extent. So this table shows to us the types of muscle fibers and the major fuel sources that is used by a sprinter and a marathon runner. So your sprinter, they make use of your type 2 or the glycolytic fibers that is being used predominantly, while in a marathoner, the predominant fibers that is used is your type 1 or your oxidative fibers. So in a sprinter, the major source of energy during the first 4 to 5 seconds is your creatine phosphate. And the glucose that is derived from your smooth muscle glycogen and metabolized by the anaerobic glycolysis is the major fuel source. In a sprinter, the muscle glycogen is rapidly depleted um, compared to your marathoner that is slowly depleted. In marathoner, the major energy source all throughout is through oxidative phosphorylation. And the blood glucose and free fatty acids are the major fuel sources when you for your marathon runner. So that's basically the end of the lecture for um, your muscle biochemistry. So when it comes to the physiologic mechanism of your contraction relaxation, that's basically part of your physiology. So those are the more important things that you need to consider when we talk about muscle biochemistry. Are there any questions before we proceed to the next topic? Okay, if there is none, let's proceed with glycogen metabolism. Okay, so um, when we talk about glycogen metabolism, no, we will try to review the importance of your glycogen metabolism and we relate the glycogen structure to the glycogen metabolism. So we will also look into the glycogen metabolic pathways. How is glycogen being regulated? And lastly, let's tackle a little bit on glycogen storage diseases. So in red cell in the brain, there is an absolute requirement for blood glucose that will be used for energy metabolism. And about 80% of the 200 grams of your glucose is consumed in the body per day. So that's basically a big amount. No? In an adult, approximately a 70 kilogram adult, um, the type that is being used in both the liver and the muscle for its metabolism is your glycogen, while in your blood and extracellular fluid, basically, you have your um, glucose stores. Now, the human body absorbs glucose from our intestines for only 2 to 3 hours following a carbohydrate containing meal. So there must be a mechanism for the maintenance of blood glucose between meals. So your 10 grams of glucose in the plasma and ECF must be replenished constantly. So when hypoglycemia develops, this will eventually compromise the brain function. This can lead to confusion and disorientation and possibly a life-threatening coma, especially when you have a blood glucose concentration that is below 45 milligrams per DL. So this is basically expected level of your hypoglycemia that can lead to neurologic manifestations. Now, your hepatic glycogen is gradually degraded between meals via your glycogenolysis, no? And this eventually maintains the blood glucose concentration. So your total hepatic glycogen stores are barely sufficient for the maintenance of blood glucose concentration during a 12-hour fast. So basically, during sleep, when we are not eating, there is a gradual shift from glycogenolysis to the de novo synthesis of your glucose, which is also a hepatic pathway known as the gluconeogenesis. But there will be a separate module on gluconeogenesis. So at least you know that there is interconnection of the different metabolic pathways in the body. So um, your sources of glucose are basically primarily from hepatic glycogen between meals. So late in the night or early morning, gluconeogenesis becomes the primary source. Your muscle glycogen is used exclusively for energy metabolism in muscle, especially during bursts of physical activity. No, because you need more energy, so you need more glucose, you need more ATP. In the cardiac and skeletal muscle, on the other hand, it rely on the fats as the primary source of energy. Some glucose metabolism is essential for the efficient fat metabolism in these tissues. So your glycogen is the principal storage carbohydrate in the body, particularly in the liver and the muscle. So your liver provides glucose for extrahepatic tissues, while your muscle mainly as a ready source of metabolic fuel for the use of the muscle itself. So take note that your muscle lacks glucose 6 phosphatase. So this is an important differentiation um, of the enzyme that is found between your muscle and the liver, because your muscle lacks glucose 6 phosphatase. So basically, glycogenesis is the production of your glycogen from your glucose molecule, while your glycogenolysis is the degradation of your glycogen to your glucose. So basically, it seems to us that it's just a reverse of the metabolic pathways, but there is a twist. No, and I know you know about it. So your CAMP integrates the metabolic regulation. Basically, this promotes the simultaneous stimulation of your phosphorylase and the inhibition of your glycogen synthase. So your insulin, on the other hand, acts reciprocally. So it inhibits glycogenolysis and it promotes glycogenesis. So there are inherited deficiencies of your enzymes of glycogen metabolism in both the liver and the muscle that causes your glycogen storage diseases. And I'll be presenting this one in the last part. No? The deposition of an abnormal type or quantity of glycogen in tissues or failure to mobilize glycogen is basically one of the mechanisms in the um, in glycogen storage diseases. As mentioned earlier, no, your in the liver, it's basically your glycogen serves as the major um, one to provide the glucose for extrahepatic tissues, while in muscle, it serves mainly as the source of your metabolic fuel. Your glycogen is synthesized from glucose by the pathway of your glycogenesis, and it is broken down by a separate pathway, which is your glycogenolysis. Okay, so can I have any volunteer to shortly discuss to us on glycogenolysis, and the other volunteer will shortly um, uh, show us through the glycogenesis. Okay, anybody from the group? Any volunteers before I call someone? Hello, are you still listening?
Yes, Doc. I
Okay,
can for the glycogenesis, we'll
sige.
start. So the glycogenesis from the term glycogen, the formation of glycogen, so from glucose to glycogen. So in here, we will start the blood glucose at the, I annotate, Doc, ha? So that
Mm-mm.
I, okay. So here, so we will start at the blood glucose. Sorry. Okay, so we'll start here from the top. So at the start, the blood glucose will interact with hexokinase for the liver and okay, hexokinase in all tissues, and but I forgot, like there's another kinase, Doc, for the muscle, but
Glucokinase.
okay, thank you, Doc. Okay, so we have hexokinase and the glucokinase. So the glucose will interact with this enzyme, which leads to the formation of a glucose 6-phosphate. So what happens here? So why there is in from the glucose, there it will become glucose 6-phosphate. So there is a phosphorylation happen. So a phosphate is added towards the glucose. So glucose 6-phosphate means that the location of the glucose can be found at the 6th the phosphate can be found at the 6th carbon. if I'm correct. And then
Yes.
with this, from the glucose 6-phosphate, since it is phosphorylated, so the function for the mutase is isomerization, or the changing of position of the phosphate from 6 place to 1 place. So it will become glucose 1
Carbon
phosphate.
1.
Yeah, very good. It will become the carbon 1. So okay, so for the carbon 1, so from the 6 carbon, it will transfer to the glucose or the phosphate for the first carbon. So it will become glucose 1 phosphate. So in here, at first, Doc, in the book, I was really confused with the pyrophosphorylase at first. So I just realized that for the glucose 1 phosphate, it will become UDP. Okay. It will become. Wait,
UDP glucose.
okay. So here, okay, here they will interact with pyrophosphorylase and UTP. So there's additional UTP here. I'll add here. Sorry, I'll add something. Okay, there's UTP here. So the UTP here is like, okay, so we have 1 phosphate here. Okay, so we have 1 phosphate here already. I mean here to here. Sorry for the wrong. We have 1 phosphate here already. So we have 1 phosphate here, and we have 2 diphosphate afterwards. So 1 phosphate and diphosphate afterwards. So what happens here at the pyrophosphorylase? So they have an interaction of uridine triphosphate. So it has triphosphate. So it has 3 phosphate. Okay, so it has 3 phosphate. So what happens here is that pyrophosphorylase is an enzyme wherein the uridine triphosphate, the two phosphate will release, and it will give 1 phosphate to the glucose. So what happens here is that there, like this, like that. So there is like okay, I'll add something. Okay, so here, so the 2-pi is released. So the 2-pi is released, and then there will be an additional phosphate towards the glucose. That's why what happened here is that there is a uridine diphosphate glucose over here. So we have glucose here. So we have glucose. And then from the uridine triphosphate, you got uridine from the uridine, and then the triphosphate here. So the triphosphate, the 2 inorganic phosphate is being released, so we have only 1 left. So we have only 1 left. So we have 1 phosphate here and 1 phosphate here. So 1 phosphate and 1 phosphate is 2 diphosphate. So that's what happens. So there will be a uridine diphosphate glucose. So I just realized that during the readings. So after becoming a uridine diphosphate glucose, okay, so there will be a glycogen synthase. So glycogen synthase, from the word synthase, I think it's synthesis of the glycogen. It will help from the glucose, the formation of bonds in becoming a glycogen. So in here, so since the glycogen synthase, the function for this one, it releases this diphosphate, it releases this diphosphate, so it releases this two diphosphate, and then what is left now is the glucose. So I think this is where like the chain, the one is 1 to 4 link chains of the linear, and it will be happening this one in this time box. So I think this is for the glycogenesis. So because of the glycogen synthase, it removes the diphosphate glucose of the glycogen, of the uridine diphosphate glucose, and only left is the glycogen. Okay. Yeah, that's
Okay.
what for.
So thank you. Can anyone go us through the glycogenolysis? Once, what happens to your glycogen? How is it being formed to glucose?
I think I can try, Doc.
Okay.
Okay. Wait. Can I annotate? Okay. So glycogenolysis starts with glycogen here at the bottom. What happens is that in creating the structure of glycogen is like they are like branched out. Like, okay, let me draw something. Okay, like circles lang. Okay, so Okay, so they are like branch chains, right?
Mm-mm.
So these branch chains are then in the case of like in hunger or there's a decrease in blood glucose,
Starvation.
there would be there would be an increase, there would be an activation of this glycogen phosphorylase. So what this glycogen phosphorylase does is that uh, it breaks the alpha 1 4 glycosidic bonds of the chain. So okay, like in what
Okay,
I
okay, I'll
draw
move you
here,
here.
the okay, yeah, thank you, Doc. Okay, here, let me erase na lang this one. Okay, so in here, what it does is that it breaks wait, this one, the alpha 1 4 glycosidic bonds. But how does glycogen phosphorylase do it? It does this by cleaving using by attaching the phosphate to the one carbon of this glucose, then releasing it. So this one becomes glucose 1 phosphate as seen here until the chain is up to only 4, wherein is another enzyme that activates here, called the glucan transferase. So here, what the glucan transferase does is that, but there's only 4 of them left. What it does is that it transfers a trisaccharide, the the tree here, into another branch. So which moves us to the next figure here. And what remains is that this one glucose that is attached to the branch by a alpha 1 6. So this debranching enzyme like cleaves this bond and releasing a separate glucose here at the right picture. It releases a free glucose. So then what happens to the glucose 1 phosphate that is being like that is like storing in the cytosol is that uh, this enzyme, the mutase, as what the previous diagram mentioned, is isomerizes glucose 1 phosphate into glucose 6 phosphate. So as what Shanika mentioned, it like changes the position. So in the phosphate in the one carbon goes to the 6 carbon, and this one is like interchangeable, is what they call interchangeable process. And
Reversible.
I have reversible, I think, and this glucose 6 phosphate is then used up in the liver cells, but in the muscle, because in the muscle it is used as ATP because the muscles can't what they call this, produce or turn this into glucose.
It because your muscle lacks. This is where the difference lies. Your muscle lacks the enzyme glucose 6 phosphatase.
Yes.
Mm-mm. So that's the difference, no, in the muscle and in the liver. So this is basically how your metabolism of your glycogen happens. So know that your glycogen storage polymer is a branched hemoglucan, and more branches are seen here than your amylopectin. So this highly branched structure is responsible for the rapid glucose release from your glycogen store, most especially in the muscle during exercise. So your glycogen rather is a core sequence that is bound to a tyrosine residue, which is your glycogenin. So the enzymes that are bound to the surface of the glycogen particle is the one that provides a ready access for the rapid release of your glucose from the glycogen polymer. Now, we already saw this earlier, how the debranching occurs in your glycogen. So basically, your glycogen phosphorylase phosphorylatically remove your single glucose residue from the alpha-1,4 linkages at the ends of the branches. So its substrates include your glycogen and your PI, and you need a cofactor that is your pyridoxal phosphate. So this pyridoxal phosphate is not the reactive group. It is the phosphate group that is the aldehyde in the pyridoxal phosphate is not the reactive group, but rather the phosphate group that is catalytically active. So in glycogenesis, now you have your glycogenine, that is a protein that serves as the core of your glycogen and is catalyzed into its glycosylation. So you have your glycan that is composed of 8 residues that eventually forms your primer. Your glycogen synthase, on the other hand, uses your UDP glucose as a substrate and then the non-reducing end of your glycogen. So as mentioned earlier, your UDP glucose pyrophosphorylase is responsible for the formation of your UDP glucose. No? So basically, we mentioned about this one early on with the metabolism of your glycogen. So what about carbohydrate loading exercise exhaustion? So di ba, you have your branch points that is being formed. This branch point formation is lower than the addition of your glucose units to a linear chain. So what happens when the muscle glycogen is largely depleted, most especially in patients who are into marathon. This is followed by um, even after a high carbohydrate meal because there is rapid glycogen synthesis with fewer branch points than the normal. So this is what happens uh, of the marathoner, no? Where there is exhaustion of your glucose because of this event. So there are regulatory parts in your glycogen metabolism. So you have your principal enzymes, which is your glycogen phosphorylase and your glycogen synthase. And this is its regulation is through allosteric mechanism as well as covalent modification. So there are regulatory hormones of your glycogen metabolism. This includes your epinephrine, your norepinephrine, your glucagon, your CAMP, and your insulin. So basically, your glycogen phosphorylase is regulated by a cascade that greatly amplifies a small signal into a very large effect. So your one molecule of epinephrine activates your adenylate cyclase, producing CAMP that helps in your glucose 1 or that activates or stimulates your glucose 1 phosphatase. This amplification system allows few molecules of your hormone leading to the depletion of your glycogen stores. So you also have your phosphorylase kinase that is responsible in the activation of the multi-subunit enzyme, which is your alpha and beta subunits that is regulatory. Here, can anyone explain to us the regulation of your different hormones as that was mentioned earlier in the glycogen metabolism? So just focus on the regulation. Can anybody explain to us this regulatory system? Any volunteer? So what is the effect of insulin? How does it regulate the enzymes that is responsible or that is part of your glycogen metabolism?
Hello, Doc. Can I try?
Yes,
I'm just gonna
sure.
base it on how I studied it.
Mm-mm.
So I'm going to talk about glucagon first. So it is known that our glucagon actually binds onto the 7 transmembrane receptors that are located in our liver cells. Excuse me for a while. Okay. So if your glucagon actually binds to your 7 transmembrane proteins located on the liver cells, on the membrane of your liver cells, they actually stimulate what we call the G protein. Remember, our G protein is actually binds to the GTP molecule. When the G protein is, pag nabind na yung GTP molecule to the G protein, it then disassociates and would attach to the adenylate cyclase. Our adenylate cyclase actually catalyzes the conversion of our ATP to our cyclic adenosine monophosphate. So cyclic adenosine monophosphate actually inactive, which actually binds to your inactive phosphorylase kinase A and would then activate it. So if it is then activated, your activated pKa would actually activate your phosphorylase kinase via your phosphorylation. So if ma-activate na siya, the active phosphorylase kinase actually activates your glycogen phosphorylase. We have already I think we have already identified that if in cases of low blood glucose levels, low glucose levels in the blood, it actually activates your phosphorylase A, which initiates glycogen breakdown. If there is high glucose level in the blood, it actually deactivates your phosphorylase A, which stops glycogen breakdown. So going back to the steps, if the active glycogen phosphorylase that would initiate the breakdown of your glycogen, hence the metabolism. So the addition of in addition to the release of glucagon, the adrenal medulla actually releases a tyrosine-based hormone, what we call the epinephrine. So epinephrine actually binds to the alpha adrenergic 7 transmembrane receptors that initiates the phosphoinositide cascade. This cascade would allow the release of calcium ions into the cytoplasm, which are needed to fully activate our phosphorylase kinase. So if ma-activate na ang phosphorylase kinase, it would actually activate your glycogen phosphorylase. So the end product of your glucagon is to promote glycogen breakdown.
And eventually increases your glucose.
Glucose, yes.
Yes. Mm-mm. So how about the role of insulin? How does it affect? How about your glucagon and epinephrine on the formation of your glycogen?
Pa rin. Ako pa rin. Ako pala.
It's okay.
Anybody?
Or any other volunteer.
Okay, I'll try.
Mm-mm.
So in cases of insulin, if you're going to ingest a meal rich in carbohydrates, the blood glucose levels actually rise above the normal value. If we're going to be specific, I think it is 5 point something millimolars during my pre-med. And this would cause the beta cells of the pancreas to produce and release what we call the peptide hormone as insulin. So now insulin actually binds to another receptor, which is named as the insulin receptor. So inside our insulin receptor, upon the binding of our insulin to our insulin receptor, it would actually activate your insulin receptor site into its active form. So your IRS actually, or your insulin receptor site is activated, which actually activates your protein kinase. Remember, our protein kinase, when in its active form, actually converts your glycogen synthase kinase, which remember, if ang active form, it would convert your it would convert your glycogen synthase that would actually promote glycogenesis. So ang insulin, ang case here is the protein kinase, the glycogen synthase kinase is actually inactivated. So the glycogen synthase that would actually ultimately cause the inactivation of your glycogen synthase kinase, the enzyme that actually keeps the glycogen synthase in the inactive form. So in order para i-correlate na po na Doc sa protein phosphatase 1, so in order to transform the glycogen synthase into the active form, thereby initiating glycogenesis, protein phosphatase 1 must be activated. Remember, protein phosphatase 1 natin, it is usually bound to a regulatory protein, making it in inactive form. So, diba, ang phosphorylase A na to, this is already a active form. This it is considered as in a relaxed state. So, nakabound dito ang regulatory protein wherein the PP1 is actually bound. So, if ever mag-bind na siya tapos there's this presence na, so ma-proceed na ma-release na niya ang attach na tong, di ba na may glucose, kitaas naman. So, kailangan mo attach ang glucose ito sa phosphorylase A nimo. When it attaches to the phosphorylase A, it actually promotes dissociation of the two kasi na nasa tense state ang imuhang phosphorylase. So if ma-separate na sila, the protein phosphatase is actually released from the regulatory protein, making it active. So what actually is the total gist is that the phosphorylase A is used to sense glucose in liver cells. So we have known that in our discussions in our SGDs that our phosphorylase B is the inactive form of our phosphorylase. It is not sensitive to glucose. That is why I said earlier that phosphorylase A is sensitive to glucose. In the active state, it is bound to PP1, right? So the association inactivates PP1. So when glucose actually binds to your phosphorylase A, it changes into the inactive nga form. So the phosphorylase B. So mag-dissociate na ang protein phosphatase 1 from the regulatory protein, then ang active PP1 now mag-dephosphorylate sa mong glycogen synthase earlier. So diba active kay it produces glycogenesis. Yeah. So it activates glycogenesis. So it is initiated by PP1. So in the total gist of it, your protein phosphatase 1 actually converts your phosphorylase A into your phosphorylase B.
Okay, so any additional info from the other students? Now, if there is none, let's proceed. Okay, so your maximum activation of the phosphorylase kinase, as mentioned earlier, requires your phosphorylation of a specific serine residues, plus the interaction of your calcium with colmodulin. So the ultimate control of your glycogen phosphorylase therefore involves a reciprocal regulation of your phosphoprotein phosphatases and phosphorylase kinase activities through your CAMP. So take note that your as mentioned earlier, your epinephrine is released from the chromaffin cells of your adrenal medulla in response to stress. So your glucagon and your epinephrine has the same regulatory function that stimulates glycogenolysis in the liver. Your epinephrine, on the other hand, stimulates glycogenolysis in the heart and in skeletal muscle. And there is a neural control that is involved in glycogenolysis, especially in skeletal muscle. And your insulin is responsible for the stimulation of your glycogenesis in both the muscle and the liver. So that's basically the gist of the regulation, no? So this was already mentioned earlier, the source of your glucagon as well as your epinephrine, no? The initiator usually for your glucagon is the presence of hypoglycemia. The same thing with your epinephrine, but as mentioned, acute stress because your epinephrine results also from that. So both hormones rapidly activate your glycogenolysis, no? So here, this is what happens during fasting and during exercise. So as mentioned earlier, you have your glucagon that is being secreted from the pancreas and your epinephrine from the adrenal medulla. So when you have a low glucose concentration in the body, your glycogen glycogenolysis happens, no? There will be metabolism of your glycogen to your glucose, no? Increasing the glucose level in the blood. So, however, when you do your exercise, what happens in the skeletal muscle is that because it uses your glucose, no, to be able to proceed with the activity, because you know when you exercise, you need your glucose for your muscle cells. So this is basically how your glucagon and your epinephrine affects your glycogen metabolism. And lastly, we have your glycogen storage diseases. So these are basically genetic enzyme deficiencies that is associated with an excessive glycogen accumulation with the cell. It can be an abnormal type or failure of the mobilization. So when there is an enzyme defect in the liver, this is basically a common symptom of hypoglycemia relating to the impaired mobilization of glucose for release to the blood during fasting. So, however, when the defect is in this muscle tissue, so this produces weakness as well as difficulty with exercise that results from the inability to increase glucose entry into glycolysis during exercise. So, additional symptoms depend on the particular enzyme that is deficient. So, I want you to go back to your book and look at again these different glycogen storage diseases. What know the different enzyme deficiencies of these different glycogen storage diseases and what happens, no? How does it present? So this is, let's not go over them one by one. So just try to familiarize yourself on this table and know the basic problem in this glycogen or the basic enzyme deficiencies that produces these different glycogen storage diseases. So I guess this will be the last part of the lecture. I hope this serves as your review for the exam on Friday, right?
Yes, Doc.
Okay. So I think that would be all. It's already 11:44. So if there are no questions, we can dismiss the class.
Thank you, Doc.
Thank
Thank you,
you.
Doc.
Thank you,
Thank
Doc.
you,
Thank
everyone.
You.
you,
Thank you,
Doc.
Doc.